For an introduction, we would like to first briefly describe what is neuraminidase. Secondly, what are the groupings of neuraminidase, and the sequences, and the structures of the neuraminidase that we are interested in, which is the N8 neuraminidase. So what are neuraminidases? Neuraminidase enzymes are glycoside hydrolase enzymes that cleave the glycosidic linkages of neuraminic acids. Besides that, neuraminidase is also a drug that is targeted for the prevention of the spread of influenza infection. It has also been targeted in structure-based enzyme inhibitor design programs that have resulted in the production of 2 drugs, zanamivir, Relenza, and oseltamivir, Tamiflu, which are currently being used to treat infected patients of influenza. Next is the grouping of neuraminidases. Neuraminidases from influenza type A viruses form 2 genetically distinct groups, which are Group 1 and Group 2. Group 1 contains the N1 neuraminidase of the H5N1 avian virus, which consist of the N1, N4, N5 and N8 subtypes. On the other hand, Group 2 contains the N2 and N9 enzymes that are used for the structure-based design of current drugs. The group also includes N2, N3, N6, N7 and N9 subtypes. Formulation of a protein drug product can be quite a challenge, but without a good understanding of the nature of protein structure and the conformational characteristics of the specific protein being formulated, the results can be ruinous. Thus, it is important for us to first identify and understand the structure of N8 neuraminidase, which is also known as 2-HT5A. Now, let us first look at the nucleotide sequence of 2-HT5A. A nucleotide sequence is any of various compounds consisting of a nucleoside combined with a phosphate group and forming the basic constituent of DNA and RNA. By convention, sequences are usually presented from the 5' to the 3' end. This is the nucleotide of 2-HT5A that is obtained from the protein databank. Next is the protein sequence, peptide sequence, or amino acid sequence, is the order in which amino acid residues, connected by peptide bonds, lay in the chain in peptides and proteins. Protein sequencing helps to determine the amino acid sequence of a protein, as well as which conformation the protein adopts, and the extent to which it is complexed with any non-peptide molecules. It is important for understanding cellular processes and allows drugs that target specific metabolic pathways to be invented more easily. This is the protein sequence of the N8 neuraminidase. Secondary structure. Secondary structure depicts the interaction of the hydrogen bond donor and acceptor residues of the repeating peptide unit. In biochemistry and structural biology, secondary structure is the general 3-dimensional form of local segments of biopolymers such as proteins and nucleic acids. This figure shows the secondary structure of 2-HT5A. Lastly is the tertiary/quaternary structure of the protein N8 neuraminidase. Tertiary structure refers to the 3-dimensional structure of a single, double, or triple-bonded protein molecule. The alpha helix fossils and beta-pleated sheets are folded into a compact globular structure. The quaternary structure, on the other hand, is the 3-dimensional structure of a multi-subunit protein and how the subunits fit together. Regarding the functions and structure of the N8 neuraminidase, there is a journal that helps to illustrate them.
The title of the journal is The Structure of H5N1 Avian Influenza Neuraminidase Suggests New Opportunities for Drug Design by Russell et al., which was published in 2006.
There are several findings from the research. The first finding is that there is a strong correlation between the extent of sequence identity and the similarity of the crystal structures. Figure shows the ribbon's representation of the group 1 and 1 neuraminidase tetramer. One monomer is colored to emphasize the molecule as a canonical six-bladed B propeller structure. In this figure, the active site region, at the center of the six-bladed B propeller structure, is highlighted, and then, shown on a larger scale in B and C. This is figure B, where superposition of the active sites of 3 neuraminidases from group 1, showing how similar they are. N1 is green, N4 is gold, and N8 is blue. Next is figure C, where superposition of the active site of N1 in green and N9 in yellow neuraminidases, demonstrating that N9 is markedly different to N1 in the 150 loop region. Conserved residues such as GLU-119, ASP-151, and GLU-276, and the hydrophobic residue at position 149, are shown in stick representation. The second finding of the research: our group 1 neuraminidases contain a cavity adjacent to their active sites that closes on ligand binding, and the result from the analysis suggests that it may be possible to exploit the size and location of the group 1 cavity to develop new anti-influenza drugs. These are the molecular surfaces of group 1 and group 2 neuraminidases with bound oseltamivir, showing the 150 cavity in the group 1 structure that arises because of the distinct conformation of the 150 loop. This is a larger view from the previous figure. The N1 in green and N9 in yellow are shown in surface representation, with the protein main chain shown in a tilde-worma representation. Next is the cavity position of the protein. Superposition of the active sites of APA N1 in green and N1 complexed with oseltamivir in blue. Part of the electron density map from a low resolution of 5.5 A difference. Fourier calculated between APA N1 and oseltamivir bound N1 datasets is shown in blue to indicate the position of the 150 cavity. In conclusion, the journal found out that Number 1. The 150 loop, which forms one corner of the enzyme active site, is able to exist in at least 2 stable conformations. Number 2. The fact that group 1 neuraminidases bind drugs, like oseltamivir, with similar affinity to group 2 enzymes suggests that the difference in energy between the 2 conformations is not very large. Next, the notion of a degree of plasticity in the structure of the active site of neuraminidase, or at least of the group 1 enzymes, is unexpected. But considering their similarities in sequence, it would not now be surprising if the 150 loop of group 2 neuraminidases was found to also possess a degree of flexibility. And lastly, the structural observations of the study found out that new drugs against group 1 neuraminidases could be obtained by adding extra substituent moieties to existing inhibitor skeletons. In any case, it may well be possible that new inhibitors designed to exploit additional interactions with the open form of the 150 loop of group 1 neuraminidases could select a similar conformation of this loop in group 2 neuraminidases.